Hello, my name is Jason McCrander. I am an associate professor of marine biology at Florida Southern College, and I wanted to go over some recent research results that we have produced here at the college in collaboration with Darrell's Aquarium, investigating something that we are both very interested in. And so the project was looking at sea anemone associations with clownfish and barriers to establishing those associations as it relates to toxin or venom dynamics. And you'll see this overview of the research, and there'll be a link to the article as well, so you could check that out there. Um, and so sea anemones are really unique because they're the only species that live symbiotically with something that they can eat, but they don't. And there's a lot of reasoning behind this, but we don't know or haven't known until now what role venom might play in establishing those symbioses, right? So one big question is, as the host that is a venomous host is in association with these potential food items or mutualistic clownfish, how do they turn off their weapons? And there's been a lot of work done in this area to look at behavioral adaptations within the clownfish to use sort of camouflage when they rub up along the anemone, they get kind of the mucus on them, and then they remain hidden among the tentacles. And so anemones are groups of this, or members of this group called cnidarians. They are one of the oldest animal lineages on the planet, and cnidarians have venom across the whole group. They use venom in a lot of different ways. So members of cnidarians are like jellyfish, clownfish, and or sorry, jellyfish, sea anemones, the corals, a bunch of different groups like that. And so one question that we had was when the clownfish arrives, do these sea anemones actually downregulate their venom and their toxins associated with it to help accommodate it? Or does the clownfish simply endure and deal with potential stings? And so to figure this out, we tracked venom expression at three different stages, which was before clownfish association, um, during that initial clownfish association, that 12-hour window after it was established, and then 48 hours or more after the establishment was taking place. And we took tentacles, clippings of the anemone at all three time points, and we compared across those time points to see how the venom expression would change. And we actually did this for five different species. Those five species represent very distinct lineages within sea anemones or organisms that have traveled across evolutionary time. And what you can see is we had three that were your typical clownfish hosting anemones that you can get from majority of pet stores. And then we also had a couple of species that were not your typical clownfish hosting sea anemones, but are actually Caribbean sea anemones. And the Caribbean does not have clownfish. And so these species have never interacted with clownfish. They don't have the adaptive traits to establish those symbioses, but they have been reported to associate in the aquarium trade. So we wanted to see, using molecular tools, how do these gene expression profiles change over time once this association is established. And we were comparing the venom association of those that are typical hosters versus atypical hosters. And what we found, contrary to what we expected, was that the overall arsenal of the venom repertoire of these species remained relatively stable. During that first 12-hour and then into the 48-hour period, there was pretty much the same venom profile during that initial establishment. Now, overall, the venom profile did not change, but there were some subtle differences, right? So the host preserves this broad defensive strategy, potentially somewhat predatory, although they rely on the symbiosis a lot for their food. So they don't compromise or adjust their venom profiles simply in response to this association. So the clownfish either remains hidden among the tentacles due to their behavioral adaptation, or benefits further from the venom that's protecting it now it's being housed. But there were some subtle shifts, and those subtle shifts could carry some significant information with regards to how venom association is established in the wild, right? So when we look at the profiles overall, they remain relatively static, but at an individual toxin basis, there were some subtle differences from one time point to the next, right? So it could be swapping out its potential repertoire components. So it's switching out different types of ammunition in response to these venom associations. And this is kind of reflected in individual toxin genes shifting their overall venom expression. Now, a lot of toxin genes occur in what's called a multi-copy gene family, where you could have different copies of the same potential venom arsenal, but there's subtle differences in those copies that might target slightly different targets, right? And so those multi-gene copies allow you to fluctuate venom gene expression from one gene copy to the next, depending on where you're going, right? And what you're targeting. And so what we saw was there could be some potential uh, deterrence for establishing symbiosis within some of these gene copies, but not broadly across the whole venom repertoire. And we saw this with Condylactis gigantea, which is the Caribbean sea anemone, and it's an atypical host. And we had not all individual sea anemones of the experiment did this, but just one. One individual had a failed association multiple times, and actually two clownfish mortalities resulted prior to establishing the symbiosis. And we didn't have the molecular data at the time prior to or during the association that was being established. We only were able to gather that after the experiment was over, but we did see one venom gene copy of an actinoporin, that was one of the most highly upregulated venom genes prior to establishment, drop down to almost nothing, and mirroring the expression profile of the other anemones after the establishment had occurred. Now, whether or not the anemone just fluctuates in their venom expression, and this coincidentally happened, or whether this was intentional decline to allow and facilitate symbiosis or potential symbiosis is requires further investigation. But this is a really good candidate to further look into, see if this targets vertebrates or has some other role compared to its counterparts. At the sequence level, it looks very similar to all other toxin genes. It had a couple of subtle differences within the protein, but for the most part, we don't know very much about what these proteins do, where the target sites are, where all the potential target sites hit. So it still is a unknown as to whether or not this carries some significant weight with the rest of the results. Um, so furthermore, whether or not this mutation carries any sort of significant role, we could further test that by isolating the protein, putting it through a series of assays to see where its activity is compared to others, and evaluate whether the fact that these mutations are absent in other described cytolytic actoporins that could carry a role. But it's a very small and subtle change. And if it does bear the responsibility of denying that symbiotic association, it warrants further investigation because that shows that symbiosis can occur with venomous animals, but there could be just small blockers or speed bumps or barriers to allow that to happen. Another thing that was really interesting from this experiment was that we had the sea anemone 8 toxin, or SA8, was very highly expressed across most of the focal species, and it doesn't really have a known a known function. And so we did see that there's uh, a lot of variation with regard to gene copy number in these other toxins. This SA8 toxin doesn't seem to have as many copies as some of these other ones, but it does have some really interesting protein regions that could bear some functional potential. Whether it is establishing symbioses or just understanding how these proteins work, there's definitely a lot more research that can be done in this area. Um, so ultimately, what we came up with was that the stable yet shifting approach really makes sense for these sea anemones because having a very diverse, non-specific venom phenotype ensures survival against predators and even protection against the clownfish. And so if they were to compromise their own venom repertoire just to allow a symbiont to establish themselves, there could be camouflage, wolf in sheep's clothing, and other potential predators getting in there and attacking the clownfish. So the clownfish has taken the right trajectory through behavioral adaptations and cloaking themselves as a sea anemone tentacle and excreting its own mucus to prevent stings. And the sea anemones don't seem to be even noticing or responding. Now, another interesting outcome that requires further investigation is there are a lot of metabolic processes that do change that affect growth and reproduction and all these other things when associated with sea anemones. And that's something that we're looking into further with this data to see what elements of metabolism are upregulated or downregulated as a byproduct of the symbiosis. So early clownfish association, the symbiosis doesn't rewrite the host's fundamental genetic defense strategy, but it could be fine-tuning it. There could be subtle shifts in individual gene copies of these venom components that are responsible. So this transcriptomic framework provides this powerful new lens for understanding how complex ancient venom systems evolve and adapt in the presence of intimate non-predatory interactions. And so we can take what we understand here and apply that knowledge to other species of sea anemones or other symbioses that occur. It's not just clownfish, it's also crustaceans, there's snails, crabs, shrimp, a bunch of things that associate with sea anemones that could eat them. So this is the first step into a long study system where we have students asking these questions, carrying out experiments, and it's just one example of the research we do here at Florida Southern College. So this project itself did take multiple years, and we have multiple student authors on it, tackling different angles and different questions associated with the project. It was quite an undertaking, and I appreciate the collaboration and support with Darrell's uh, aquarium shop with facilitating the acquisition of some of these animals used for the research. Thank you very much. And if you have any questions, please feel free to reach out. You can find me on the Florida Southern College website.